Thank you, Scott. So my name is Raud Razzaghi.
And I'm Dania Keller, and our project was the effects of creatine incubation on skeletal muscle function.
So first, a little bit of background on doxorubicin, or dox for short. It's an anthracycline antibiotic, meaning it's a chemotherapy drug used or that's engineered to either stop or severely slow down the growth of malignant cancer cells. It's used for a variety of cancers, most notably leukemia, which is blood cancer, Ewing sarcoma, cancers of the head and neck, lung cancer, liver cancer, and pancreatic cancer. And it's also used for some diseases like AIDS.
So a little bit more on doxorubicin. It creates free radical damage, and free radicals are extra electrons in the valence shell, and it tries to grab electrons from the nearest thing. So when it gets in the cell, it can create damage to the DNA or create oxidative stress, damages the DNA, the proteins, or the lipids in the cell membrane, causing cell damage or cell death. And when it kills the cells, it decreases muscle function, so it decreases the quality of life of the patient.
So now a little background on creatine. It occurs naturally in the skeletal muscle of most vertebrates and all mammals. Okay, so it's important for energy metabolism in cells. So as you can see, when the muscle contracts, ATP, adenosine triphosphate, loses one of its inorganic phosphate groups, becomes ADP, the used form of the energy. So creatine, it helps to naturally increase the levels of phosphocreatine, and phosphocreatine has a phosphate group, so it can bind to the ADP and create ATP again, so it can replenish the energy source of the cell. And also studies have shown that overall creatine can help increase muscle strength, it can help increase muscle growth, and also muscular performance.
So our research purpose was to investigate the extent to which creatine can reduce free radical damage in skeletal muscle dysfunctionality that is caused by the doxorubicin. So basically, since creatine can strengthen muscles and can act as an antioxidant, and doxorubicin damages muscles and creates oxidative stress, and what we were hoping to see from our research was that the creatine would prevent these harmful effects done by the doxorubicin.
So now a little background on the soleus and EDL muscles in rats. So the soleus is a type 1 muscle, meaning it's composed of mostly slow twitch muscle fibers. So it has a low energy output, but it tires slowly. So it's good for endurance exercise. The extensor digitorum longus, or the EDL for short, is the exact opposite. It's a type 2 muscle, meaning it's composed of mostly fast twitch muscle fibers, so it has a high energy output and it tires quickly. So this is not a rat leg, it's a human leg, by the way. So we decided to use the soleus and EDL muscles because they have similar functions in both rats and humans. In the soleus muscle in rats and humans, which soleus is right here, this one. The soleus is used for standing, so you can see why it would be a slow twitch muscle. It's used for endurance because you can stand a lot. That's for rats and humans. The EDL in rats and humans is on the other side. It's right here. It's used for functions like jumping. And most skeletal muscles in the body are a combination of the two muscle fibers. But these are almost completely their own type of.
Okay, so for our method, we had 3 different buffer groups: the Krebs, the creatine, and the dox. And we used 2 stag dolly rats for each group. So our first group was a control group, which is the Krebs buffer, and then we switched it into another Krebs buffer. And the Krebs buffer is made of things to make it very similar to the how it is in the body, to keep it as realistic as possible. And our next group was the Krebs to doxorubicin buffer. So ideally, at the end of this treatment, the muscle function would be at the lowest. And the next group was creatine to Krebs. So that should ideally be the highest functioning, because the creatine builds up the muscle strength. And then our test group was the creatine to doxorubicin. Hopefully, or we were hoping it would be about the same as the Krebs to Krebs functionality, or at least higher than the Krebs to doxorubicin.
Okay, so all animals were treated in accordance with the IACUC, and they were treated humanely. So we worked with one rat at a time, and when the rat was ready for use, we anesthetized it with 0.7 milliliters of sodium pentobarbital, which is a euthanizing compound, and 0.3 milliliters of heparin, which is an anticoagulant. Once the animal had passed, we removed the EDL and soleus muscles surgically, and we placed them on this muscle function apparatus. It looks pretty complicated, but it's not that complicated. This right here, you can't. Well, that's the reservoir at the top, a glass thing. That's where we put the buffers. And then the buffers would flush into these 4 baths, which were where we put the muscles. You can kind of see there's metal electrodes there. That's how we stimulated the muscle itself. When we took the muscle out, we attached it to strings and put it on these hooks right here, and we incubated it in the buffers, which were in these baths. So we could mess with a few of the settings on the muscle function apparatus. We could play with the tension that we put on the muscle, and we could also play with the voltage that we stimulated the muscle with for it to contract. So once we played with those 2 settings, and once we attained the maximum twitch contraction from each muscle, then we started the 5 minutes for every 30 minutes procedure. We stimulated the muscle at those settings for every 5 minutes for half an hour. Then we flushed out the first buffer and put in the second buffer into here, so that the second buffer, whichever it was in that particular group. And we stimulated the muscle again every 5 minutes for half an hour. And then we started the exhaustion protocol, which was where we constantly stimulated the muscles until exhaustion. So that's supposed to simulate fatigue.
So the second part of our research was to the myosin heavy chain analysis. This was to isolate the protein carbonyl, which is a marker for oxidative stress. So it would mark the damage done to the cells by the doxorubicin. So after the muscles were tested on the muscle function apparatus, they were frozen. So we thawed out the muscles, and we had homogenized them with a myosin heavy chain buffer. And after that, we centrifuged them down to separate the heavy protein. So we'd have a supernatant and the pellet. And the pellet was the heavier protein, so we would dump out the supernatant and wash it and resuspend it. And then we added the Bradford reagent, which changed the color of the proteins, and put it through a liquid spectrometer, which would measure the color of it and give us the exact protein carbonyl concentration. Okay. So for our results, on the x-axis we have time, and the y-axis we have force of the muscle contraction. As you can see, the black line is the creatine to dox, which was the test group, and the red line is the Krebs to dox, which was the lowest one. And we did find a significant difference between the 2 lines, which showed to us that the creatine did help fight against the harmful effects of the doxorubicin. And to show a little more, the green line was the creatine to Krebs, the highest group, which showed us which showed us that the creatine does strengthen muscles. It is the highest line on there. Next, we have the soleus muscle graph. We did the two graphs to show the difference between the two muscles. Again, the creatine to dox and the Krebs to dox had a significant difference, showing that the creatine had a positive effect.
Okay, so this is a bar graph of the protein carbonyl formation in only the soleus muscles. So again, protein carbonyls are the standard marker for protein oxidative stress. So the more protein carbonyl concentration there was in a muscle tissue sample, the more protein or the more oxidative stress there was, the more free radical damage had occurred. So the protein carbonyl concentration is measured in nanomole concentration, and so we can each bar graph. So each muscle, each rat had two soleus muscles, one in its right leg, one in its left leg, and because we had two rats per each group, each bar represents 4 soleus muscles. So we can see that the creatine Krebs group had the smallest concentration, the lowest concentration of protein carbonyls, meaning it had the lowest level of oxidative stress, and the creatine dox group had the highest level of protein carbonyl content, meaning it had the highest oxidative stress. And the creatine dox group, which is our experimental group, you can see that it has a significantly less, or it has a lot less protein carbonyl content than the Krebs dox group, which shows that it can help reduce the dox-induced cardiotoxic, or not cardiotoxic, dox-induced protein carbonyl, dox-induced oxidative stress. So, and it is a significant difference between, a statistically significant difference between these two. This bar graph is a total protein carbonyl formation graph. So we use both the soleus muscles and the EDL muscles. So 4 soleus, 4 EDL muscles in each bar graph, 8 muscles total per bar. However, some of the EDL muscles did not survive the surgery, so that's why we had to account for it. We accounted for that with the error bars. So we can see a lot of similar trends between the two graphs. The creatine Krebs group had the lowest level of oxidative stress, and the creatine dox group had lower than the Krebs dox group. This still is a significant difference between the two. However, the one difference you can see between the two graphs is that the Krebs group in this one had a higher concentration of protein carbonyls than the Krebs dox group here. Oxidative stress does occur naturally because of environmental factors and metabolism, but it's highly unlikely that more oxidative stress occurred naturally than that was produced artificially by the doxorubicin. So we think this might be a little bit inaccurate. So we'd like to make a few acknowledgments. First, a huge shout out to our genius prodigy mentor who's just sitting back there. His name is Eric Bredal. He responds to Eric, and he also responds to the great, incredible Hulk. Thank you to David Hydock, Dr. Hydock, who let us use his lab. Thank you to the Newmont Mining Corporation for sponsoring my attendance at FSI, and the Edward Madigan for sponsoring my research.
I would like to thank Excel Energy for sponsoring my research as well. And we would both like to thank the Frontiers of Science Institute staff and the University of Northern Colorado.